This is LA Theatre Works. I'm Susan Lowenberg. I'm joined now by Brenda Maddox, the world's foremost biographer of Rosalind Franklin. Miss Maddox has written acclaimed biographies of William Butler Yeats, Nora Joyce, and D.H. Lawrence, for which she was awarded the prestigious Whitbread Biography Prize. She has written for the New York Times, the Washington Post, the Times of London, and the Observer, among others, and regularly contributes to BBC Radio 4. Brenda Maddox, welcome to LA Theatre Works, and thanks for joining us.
Thank you.
Most people who know of Rosalind Franklin have a particular image of her in mind. Could you describe the stereotypical Rosalind Franklin?
Well, what they know of Rosalind Franklin comes largely from James Watson's book, The Double Helix, in which he described her as a kind of bad-tempered blue stocking who wouldn't let other people see her work. His portrait of her has been much criticized, but my answer to that is that if it hadn't been for Jim Watson, nobody would have heard of Rosalind Franklin.
As her biographer, do you agree with his assessment and his description of her?
Yes, I think she did annoy a lot of people, and she did have a fierce temper. What I don't agree with is the kind of feminist myth of her as the wronged heroine. She was a very good scientist, and she didn't like the lab she was working at, and she wanted to get out as soon as possible. So it was as much her fault as anybody else's that she wasn't there to profit from the great discovery that Maurice Wilkins, Francis Crick, and James Watson made with the result of her X-ray photographs. She was so unhappy at King's College London that she moved to a much less prestigious college within London, Birkbeck College. So her work, which she had left behind, was shown to Watson and Crick, and they went on to one of the greatest scientific discoveries of all time. But that's why she wasn't there.
Describe for us Rosalind Franklin's early life. What sort of a life did she and her family have as Jews living in England?
She was a type which is probably not too well understood in America, but very affluent, upper-class Jews who had come to England in the 18th century. They were very well established as bankers, and they tend to marry each other. It was called the cousinhood. She didn't marry anybody, but not because they were Jewish, but just because she was just so interested in her own work. But they lived in Kensington, North Kensington, in a very good part of London, and she went to St. Paul's Girls' School, and then to university. She was just very good at everything she did.
Was Rosalind encouraged in her mathematical interests as a child?
Her family respected her intellect. They just let her get on with it. I think they didn't push her in any way. I mean, she had a very good early education, and she was very good at it. And there was none of this sense one thinks to think that people held girls back in the past. Her family certainly had absolutely none of that. She belonged to a family which respected women's achievement.
Rosalind went to Cambridge in 1938. Was this decision supported by her family?
Oh, yes. They certainly wanted her to get on. They were kind of a family that wanted the best of everything for their children, and you couldn't get better than Cambridge.
How would you characterize her years at Cambridge?
I think she was very, very happy there. She did love science, and she loved getting on with her work, and she, as far as I know, had no social problems. She learned a lot, and she profited from it completely.
Rosalind relocated to France several years after graduating from Cambridge. What brought her there, and what sort of work was she doing?
She had a good job in a lab, and she hadn't been able to find a job in London that would actually allow her to do the kind of research she wanted to do. So she was working very happily in Paris, but her letters make clear all the time that she wanted to come back to London. She was looking for jobs at the Royal Institution, at King's College, at Birkbeck College, and when she finally got an appointment at King's College, she immediately came home.
Was Rosalind brought to King's to work specifically on DNA?
No, J.T. Randall, who brought her down, he said he wanted her to work on certain acids in which we are interested. He knew from her French work that she was very good at X-ray photography. And it just so happened before she got to King's, he got some wonderful samples called Signer DNA, which was very, very good for photographic purposes. So when she got there, he gave her the material, and it was just perfect for the work she knew very well how to do.
Did Rosalind have any sense that her work, after she left King's, was being co-opted by Watson, Crick, and Wilkins?
No, she didn't. To her, she was working on something different called RNA, and those were exciting initials as DNA. And she died of cancer, unfortunately, sadly, in 1958, before Watson, Crick, and Wilkins got the Nobel Prize for the discovery of the double helix of DNA. But she didn't have a sense that, oh, they've stolen it from me. She was more interested in what she was doing next.
So how close was Rosalind to uncovering the true model of DNA on her own, even though she may not have realized at the time the importance of what she was doing?
Probably, it has been said, a couple of months away. If she'd persisted at it, she would have seen it. But also in science, you have to share knowledge with other people, and she was too much of a loner. I think that really held her back. But if she had, if science is full of ifs, if she had stayed at King's and she had realized, and she had talked to Watson, who's coming down from Cambridge, interested himself in trying to figure out how DNA replicated, she might have got it, but it's just what might have been.
So in a way, her own limitations, her inability to truly communicate with people, is what negatively impacted, the best outcome of this research and all this work she'd done. I mean, in a way, she sort of threw it away without knowing it.
She did, without knowing it. But remember, this was 1951. And nobody, it wasn't like the North Pole or something, people striving out for a known target. They didn't know quite what they were looking for, and they were just fumbling around. And she was a wonderful scientist at a time when there weren't many women scientists. But she wasn't striving for a goal, and certainly not for a Nobel Prize.
How was Rosalind memorialized at the time? Was there any controversy over her legacy in the immediate aftermath of her death?
Nobody had thought of her until James Watson's book. And Watson's book came out in 1968. And of course, by then, feminism had got underway, and people started to say, where are the women scientists? Why is their work not being recognized? But she just died almost too early, before people began to expect that women even did science.
Did any of her former collaborators come forward to set the record straight, as it were, about her true contributions?
Well, I think after Watson's book came out in 1968, she died in 1958. And as her family said, I'm glad she never lived to see the book, because she probably wouldn't have written so frankly. And yes, Don Casper has, and even Maurice Wilkins, various people have acknowledged, and of course the feminists have taken up the cause and really tried to restore her or reinvigorate her reputation.
What sort of a model or influence does she serve for young women scientists working in the field today?
She's an inspiration for young women, that women can go into science, and they can even now, they're learning, go into physics. I think today's young women can't imagine what it was like all those decades ago when, women weren't taken seriously, luckily, because if they're interested in science, they will do it, and without thinking, I can't do this because I'm a woman.
Brenda Maddox, thank you for speaking with us today. This was very interesting.
Thank you.
I've been speaking with Brenda Maddox, the author of Rosalind Franklin, The Dark Lady of DNA. This is LA Theatre Works. I'm Susan Lowenberg. Joining me now is Dr. Pamela Bjorkman, the Max Delbruck Professor of Biology at the California Institute of Technology and an investigator at the Howard Hughes Medical Institute. Dr. Bjorkman is the head of the Bjorkman Group at Caltech, where she uses X-ray crystallography and biochemistry to study immune recognition in proteins, particularly as it relates to HIV. Dr. Bjorkman was named the 2006 Laureate for Women in Science and was elected to the U.S. National Academy of Sciences in 2001. Dr. Bjorkman, welcome to LA Theatre Works, and thanks for joining us.
Thank you. It's great to be here.
Let's start with a basic question about your field. What is X-ray crystallography, and why is it important?
X-ray crystallography is an imaging technique that gives you a picture. In this case, it's being used for biomolecules. It's analogous to microscopy. So in microscopy, you use some form of radiation, for example, light or electrons, and this radiation would interact with a sample, and then it would be refocused to give you a magnified view of the sample.
So is this like a sonogram, except that it's taking pictures of things we can't even see?
There are some technical differences. A sonogram would give you directly an image, whereas because there's no lens that would refocus X-rays, you have to do mathematical computation to actually get the image out. But in a sense, it's giving you a view of something you wouldn't normally see. In a sonogram, it would be, say, the inside of your body. In X-ray crystallography, it would be a molecule that you couldn't possibly see because it is hundreds of millions times smaller than anything you could hope to see.
What are some of the key applications of X-ray crystallography?
Well, it's used today for many applications in biological and biomedical sciences, as well as in geology and material sciences. But in the biological field, for example, drugs such as the antiretroviral drugs that are being used to treat HIV were designed by fitting them computationally to the three-dimensional structures of viral enzymes that were derived by X-ray crystallography. And three-dimensional structures of proteins can be used to understand the function of these proteins, like how they recognize DNA to turn on or off gene expression, or how proteins can function as motors to move along microtubules in the cytoplasm of the cell, or how viruses recognize their receptors on cells, or maybe how antibodies can bind specifically to viruses and to other pathogens that they encounter.
How important is Rosalind Franklin in the field, and why?
I think Rosalind Franklin is becoming increasingly important to the field of structural biology today, now that her story is becoming more well-known through plays such as Photograph 51 and books such as Brenda Maddox's book, The Dark Lady of DNA. So Rosalind Franklin was a woman who made truly fundamental contributions to three different fields. First, her studies of the structures of coal and graphite, which she conducted during her doctoral work and later in France. And then second, her DNA structural studies, which is what we'll be discussing today. And third, after she worked on DNA, she worked on the structures of viruses. I think it would be remarkable if she'd produced important results in only one of these fields over the course of, say, a normal scientific career. But she actually made fundamental contributions to all three of these fields before her death in 1958 when she was only 37. And she did all of this despite what I would say would be the less than hospitable atmosphere for women in science that prevailed at that time. So for me, Rosalind Franklin has always served as a role model for how a scientist can keep on working on a difficult scientific problem because they really want to know the answer, not because they want potential glory or recognition or for being first in a so-called race. And one thing that the Photograph 51 play emphasizes is that Rosalind Franklin was not interested in quote-unquote racing for the structure of DNA. She was really driven by the science, not by competitiveness or, say, the desire for recognition. And I think the best science is done when scientists really wants to know an answer because of his or her own interest and love of the subject, not because they're in a race for a prize.
Could Watson and Crick have made their discoveries about DNA without Rosalind's groundwork?
I think there's really no question that Watson and Crick relied on Rosalind Franklin's data to formulate their model for the structure of DNA. So not only did her data prove that DNA was helical, it gave Watson and Crick the distances and other parameters they needed to start the model building that they did. So the play highlights the importance of the photograph 51, which is this famous picture that shows the X structure. So it highlights the importance of that image for Watson and Crick's discovery. And that's, of course, accurate, but Watson and Crick also had access to a report that Rosalind Franklin had written for a biophysics committee visit to King's College, and that report included summaries and some of these critical parameters of her results. And without these data, not just the photograph, but also this report, there would have been no basis by which Watson and Crick could have built a model. So you might ask the question, could someone other than Rosalind Franklin have recorded the necessary data to deduce a structure for DNA? So I think the answer to that is yes. And in fact, Maurice Wilkins had started X-ray diffraction studies of DNA fibers at King's College with Raymond Gosling before Rosalind Franklin arrived there. But Rosalind Franklin was really an unusually gifted experimentalist and an excellent theoretician. And the quality of the images she produced and her interpretations of them were really critical to getting the double helical model of DNA. So to answer your question simply, no. Watson and Crick could not have made their discoveries about DNA without Rosalind Franklin's data. In my view, it would have been fine to use her data for building the DNA model had she known it was being used and had she given permission for its use. And under accepted scientific codes of conduct, she would have been a co-author on the Watson and Crick paper.
The play Photograph 51 focuses on Rosalind's work on DNA. What specifically did Rosalind Franklin discover about DNA that was so revelatory?
So to answer this question, I first have to review the structure of DNA and why it's important. At the time she started her DNA studies in the early 1950s, DNA was believed by most scientists to be the biological macromolecule that determined heredity. That is, it was the genetic code that's passed on to descendants to determine their basic characteristics. So it would make a horse, or a person, or it would make one person different from another person. So the basic question to be addressed about DNA was how can it be copied in order for genetic instructions to be passed along to descendants? And also, how was the code in DNA translated into proteins? Because proteins are really the molecules that carry out the cellular functions. DNA is the information. So DNA means deoxyribonucleic acid. So that means it contains a collection of deoxyribose sugars that are connected through a phosphate-containing backbone. So at the time that Rosalind Franklin was working on the DNA structure, it was understood that DNA had 4 deoxyribonucleotide bases, adenine, thymine, cytosine, and guanine, that are abbreviated A, T, C, and G. So we now know DNA as the famous Watson and Crick double helical structure, in which the sugar phosphate backbone is on the outside, and then the 4 nucleotide bases are on the inside, and each of the bases is interacting with a partner on the opposite strand, so that A is always paired with T and G is always paired with C. So now I can finally answer your question. What Rosalind Franklin figured out through her X-ray imaging of DNA fibers is that DNA was helical, and she determined the parameters of the helix. For example, the distance between phosphates, the radius of the helix, the distance between each turn of the helix, and so on. And from the water content of these DNA crystals or fibers, she deduced that the phosphate groups were on the outside and the bases were on the inside of this structure. And this is really critical information that led to the double helical structure that had internal base pairs.
Describe for us the process that Rosalind used to arrive at her X-ray images of DNA.
Well, she used fibers of DNA in which millions of strands of double helical DNA are aligned with each other, and then they create a fiber. And then the fiber is placed in the X-ray beam, and this causes X-rays to be scattered in different directions. The scattered X-rays were then recorded on photographic film, and unlike imaging of samples using a light microscope or, say, in a sonogram, there's no way to refocus the radiation to obtain an image. There's no lens that will refocus these scattered X-rays, so the scattered pattern of X-rays has to be interpreted computationally to calculate a three-dimensional structure.
After her work at King's, she started to work in other areas of science, which you talked about a little bit before. Can you elaborate on what she did after she left King's and what that contribution was?
Yes. She moved to Birkbeck College in 1953, and she was using X-ray crystallography to study viruses. And one of these was tobacco mosaic virus. This is a virus that infects plants, and it uses RNA as its genetic material. So viruses are simple organisms, and they often consist of just a protein coat or shell that surrounds the genetic material, and the genetic material can be RNA or DNA. And viruses can afford to be very simple because they hijack cellular machinery within the host cell that they've invaded, and this is in order to make copies of themselves that can go on to infect other cells. So the reason that it's important to study viruses is to understand how they can encode enough information in such a simple structure to actually carry out this hijacking of the cellular machinery, how they infect other cells, and so on. So tobacco mosaic virus served as a model virus for understanding other pathogenic viruses, say that infect people or animals. So it turns out that the structures of simpler viruses are really beautifully symmetric arrangements of protein shells. For example, TMV is a rod-shaped virus, and the coat proteins assemble into a helical structure around the RNA.
And can you explain to our audience and me what RNA is as opposed to DNA?
Okay, so RNA is ribonucleic acid instead of deoxyribonucleic acid, and it's a simple chemical difference from DNA, but functionally what it is it's the intermediate. It is the messenger that DNA is transcribed into, from which protein is translated. So in the famous words of Francis Crick in The Central Dogma of Molecular Biology, Crick said that DNA makes RNA. So DNA is transcribed into RNA, which is a messenger, and then RNA is translated into protein. So RNA is another nucleic acid that's an intermediate between DNA, which is the information code that's passed on to descendants, and protein, which is actually the functioning molecule that carries out metabolic events and so on inside cells. So if you're a virus, you have to carry genetic information. You can carry that in the form of either DNA or RNA. And tobacco mosaic virus happens to carry RNA. Other viruses, such as polio virus, which Rosalind Franklin worked on also, the cause of polio, to which we have a vaccine against today. These viruses are RNA viruses as well, and they have icosahedral protein shells that are basically miniature geodesic domes. They're very beautiful structures. So Rosalind Franklin made many important contributions to the structural biology of tobacco mosaic virus and icosahedral viruses such as polio, and in particular defining the role of symmetry in the architecture of these viruses.
How has X-ray diffraction advanced since Rosalind's time, and what are the major developments in the field?
Well, certainly if people want to study the structures of DNA and the proteins that recognize them, they would conduct X-ray diffraction experiments using crystals of DNA, pure crystals, instead of fibers. And there are technical reasons that it is easier now to do that with crystals. So in fact, it wasn't until 1980 that the Watson and Crick model for the structure of DNA, which was deduced from the X-ray scattering from DNA quasi-crystals or fibers. It wasn't until 1980 that the Watson and Crick model was confirmed by Richard Dickerson, and he used direct experimental techniques using X-ray crystallography rather than the scattering from the DNA fibers. So now it's possible to use X-ray crystallography to look at structures of very large complexes of DNA with proteins bound, for example, proteins that regulate transcription. And it's also possible to study the structures of certain types of intact viruses, such as poliovirus, that Rosalind Franklin had to study with more indirect means.
Do you still use any of the techniques pioneered by Rosalind Franklin?
Well, the field's certainly advanced in a great many ways, including having automated detectors that are digital that read out the intensity of the scattered X-ray beams. Most of our work is done in a more automated way than what was perhaps depicted in photograph 51 with developing films and looking at them. We have a lot more use of computers, which are a lot faster and that can do a lot more calculations. Many of the calculations that Rosalind Franklin was shown doing by hand, for example, an important calculation that she was doing on the A and B forms of DNA called a Patterson function, these are all done using computers now, not by hand. But certainly we use some of the techniques that she was using. It's the same basic experiment. You put, in this case, a crystal into an X-ray beam, and then you interpret the pattern of scattered X-rays.
How accurately does the play Photograph 51 depict the hard science?
Well, I think it's hard to portray what really goes on in a lab, in a play or a movie, because much of a scientist's typical day would involve experimental manipulations or maybe technical conversations with other scientists, and that wouldn't make for, it would be kind of boring. So it wouldn't make for good dramatic theater. But with some artistic license, I think the play does a good job in making the science interesting. And for the most part, it's accurate. Now, I had a small issue with one part of the play in which Watson and Crick come to a sudden understanding of how DNA is copied after they figure out that the structure is an antiparallel double helix. That is, the strands of each of the helical parts run in opposite directions. But they come to this understanding before they figure out the specific pairing of the bases. That is, A pairs with T and G pairs with C. And it really is the base pairing that leads to an understanding of how one strand can serve as a template for another strand so that you can copy the genetic information. It isn't sufficient that the structure is an antiparallel double helix. In other words, it's necessary that it's antiparallel, but it's not sufficient. And I think this is a minor point. What may be more significant, however, is that Don Casper, who is portrayed in the play as being at King's College during the work on the structure of DNA, he didn't actually meet Rosalind Franklin until after she'd left King's. But he has made the point that Rosalind Franklin would not have exposed herself to the X-ray beam in the ways that are depicted in the play. So he doesn't believe that exposure in the lab to X-rays had anything to do with the ovarian cancer that eventually killed her.
That's very interesting, because I think a large point is made about that. I mean, you get the clear indication that that was a cause. Are there any details that you think are central to the story that the play leaves out?
Well, I think in general, the contributions of Francis Crick to the discovery of the DNA structure and the force of his personality, let's say, and his incredible intellect, I don't think these come across in the play. I never actually met him, unfortunately, but he's been described to me as amazingly dynamic and brilliant, and certainly he continued to make fundamental contributions to science long after the structure of DNA. And in the play, he sort of appears more in the role of Watson's sidekick. And I actually had the pleasure of watching the play at the World Science Festival in New York City last June when Don Casper and Raymond Gosling were in the audience. And I mean the real Don Casper and the real Raymond Gosling. And they both said that it would be difficult to impossible to capture the essence of Francis Crick in a play. So it's hard to know how his character might have been changed in the play to be more accurate. Regarding the science in the play, I really think that a critical thing that led to the Watson and Crick model for the structure of DNA that isn't depicted in the play is the whole concept of base pairing. I mean, this is mentioned in the play, but the way it was discovered is actually interesting. It resulted from Jim Watson sharing an office in Cambridge with Jerry Donahue. He was a chemist who'd gotten his PhD at Caltech, my institution, with Linus Pauling. And Donahue told Watson that the conformations of the nucleotide bases that were being depicted in textbooks at the time were likely to be wrong. And he suggested to Watson that he should try modeling the bases in their alternative conformations. And in the alternative conformations, it was then clear that A could pair with T to make a base pair, that was the same size as the base pair when you paired G with C, and then you could fit these base pairs inside the double helix. And the whole concept of base pairing explained what Jim Watson wanted to understand, which was called Chargaff's rules. This refers to a discovery by a chemist named Erwin Chargaff that if you measured the amounts of A, T, C, and G in a cell from any organism, you'd find that the amount of A would be equal to T and the amount of G would be equal to C. And so the base pairing actually explains those equalities. And as I emphasized before, the hypothesis of base pairs, it really is the critical aspect of the DNA structure that explains how it is copied.
Well, this has been very, very interesting, Dr. Bjorkman. Thank you so much for speaking with us today.
Thank you. It's been a pleasure.
I've been speaking with Dr. Pamela Bjorkman, the Max Delbruck Professor of Biology at the California Institute of Technology.